So on to topic, the second half of topic 5.4, non-Mendelian genetics, things that kind of go against, or not quite go against, but things that are maybe a little bit different than not as simplistic as Mendel believed they were in his time. So first, before I move on to the next topic, I want to review, and please pay attention here because it's going to be crucial. I want to review kind of the basics of meiosis here, right? So we have a hypothetical individual here, who is a little a, big B, little b, same individual over here on the other side. And when we talk about the gametes that are made, when we talk about division, we know that he's either going to give up a big A and a little a, or a big A or a little a, and a big B or a little b. Same with, and I say he, but he or she. Same with this person over on the other side. They're going to give a big A, little a, big B or little b. Now, this is just simply two different situations we could be in, right? Maybe the two genes we're dealing with, maybe it's eye color and hair color, something like that. Maybe they're located on different chromosomes. In this case, our B gene, maybe this is our eye color gene, is over here on this small chromosome. And the big A gene over here, maybe that's hair color, is located on this large chromosome right here. Of course, one's blue, one's red. Get one from mom, one from dad. Of course, this is unreplicated at this point because it's just for simplicity's sake. We haven't replicated this DNA. We're just showing these chromosomes as one single unreplicated strand here. So as we know, there's a 25% chance that we end up with each of these situations. Big A, big B, little a, little b, little a, big B, and big A, little b, right? Because these chromosomes, or these chromatids rather, are going to are separate, and these alleles are going to separate from each other, right? So that's something to keep in mind. Also, if you have genes far apart on the same chromosome, you are also going to have a 25% chance of each of these situations occurring because crossovers are so frequent between these two, since there's such a large space here for crossovers to occur, that they're effectively like being on separate chromosomes, right? When these segregate into their separate gametes over here, you're going to see you get about a 25% ratio of each of these as well. So genes on different chromosomes, genes far apart on the same chromosome, you end up with 25%, 25%, 25%, 25%. That's why we can make these nice, these 16 square Punnett squares. I know it's real tiny, but, and we go, oh, big A, B, big A, little b, little a, big B, and little a, little b, right? We can make these squares like this and say stuff about probability because we're assuming that each of these have a 25 probability of forming, right? Each of these gametes has a 25% probability of forming. Now, that's not always the case. And the next thing we're going to talk about is something called genetic linkage. Genetic linkage. And we're going to be talking about genes that are linked together, right? And I've kept this up here in the corner so that we can kind of compare how this happens. Instead of doing genes far apart on the same chromosome, I want to do genes near each other on the same chromosome. So let me go ahead and draw two chromosomes over here. I'm going to make them nice and big. That way I can tell. These are homologous chromosomes that code for the same stuff. Now let me locate these genes here. We're going to say that big A is right here. And we'll say this guy's heterozygous as well, same genotype for this person. And now I'm going to put these really close together. This is big B is the second one here. Put them really close together here. Let it be right there. Actually, I'm going to try and draw these on the same side because I might be using this. So let's put big B as this second one right there. Little b is right there. So as you can tell, these genes are very close together, right? And also, that's an arrow, that's not an A. I hope you guys aren't confused there. It's hard to draw. Oops. Hard to draw this so small, but there we go. Okay, so these genes are right next to each other on the same chromosome. Now, as you know, these things are going to segregate into their, into these same distinct possibilities, right? We could have big AB, big A, big B, little a, little b. We could have little a, big B, and we could have big A, little b, right? These are our same possibilities from up here. And they're also going to happen, right? This is something that also is likely going to happen. The only difference in this case is we have to take into account crossing over, and how that frequency can change. So what do I mean by that? Well, it's important to keep in mind that crossovers, right, between two separate chromosomes. Let me draw these chromosomes out over here, right? Draw some long, thin chromosomes out over here. I know they don't look the best, but bear with me. Crossing over is going to happen, right? This is just something that's going to happen. And what is going to happen is you're going to have a swap of genetic material somewhere on these chromosomes. In other words, this red piece over here and this blue piece over here from these two chromosomes are going to swap places, and you're going to end up with some unique looking chromosomes. So again, this is unreplicated DNA, but if we pretended that this thing replicated, right, and you had two X's here, crossovers can happen anywhere. However, what is the likelihood of getting a crossover that occurs? And I want you to pause the video and just look at the screen for a second. And I want you to think about this. What's the possibility of a crossover happening right in this specific zone right here, right? Because that's the crossover you would need in order to get, let's see here. Let's say you have, I'm going to draw some of these down here right now. That way we can kind of see here. Big A, big B. And this over here is little a, little b, right? Now, in order for us to get a little a, big B sperm out of here, we need a crossover to take place right in between here. And what do you think? Is that going to be very likely that you have a crossover occur between these two genes that are very close together? And the answer to that is no. It's going to be much less likely. So let me draw some of these over here, as you might see these what's called a recombinant chromosomes over here. Let me let's do this right here. So actually, let me swap this. Let's do blue here, blue up this way. Apologies. Okay, so over here I've drawn the little a and big B, and then big A and little b. These are our recombinant frequencies here, or recombinant chromosomes here. So my question to you is, are we still going to get 25%, 25%, 25%, 25%? And the answer to that is, in this case, no, we won't get 25%. We will not get 25% of each of these because these genes are linked. We might even get something as drastic as, let's say 49% of these gametes, 49% of these, and then 1% and 1% if they're close enough, right? What this means is you're going to see a lot less of these specific gametes. These are traits that travel together. You can think of it as maybe something like red hair and freckles. Again, that's that. I'm not claiming that is a trait that travels together, but I am showing you that sometimes you do see traits that seem to be similar, right? They seem to kind of travel together, as it were. So these are called linked genes or genetic linkage. When you have genes that are so close together that these crossover events occur extremely rarely between these genes, splitting them up, right? So that is genetic linkage. And if you want, take some time to kind of look at the difference between what I've drawn here and what's going on in the corner over here, so you can tell why you don't get these nice 25%, 25%, 25%, and 25% ratios for each gamete. Okay, so the last topic for non-Mendelian genetics I wanted to mention, of course, this isn't really topic 5.4. It's really 5.3, but I put this back here because I like to show this, and this is, you might remember, this is a pedigree. This is a pedigree. You might remember this from freshman year. I like to show this because it really addresses all of these different situations. And you do need to kind of understand non-Mendelian genetics to know how stuff is inherited in a pedigree, right? So first off, what I do when I look at a pedigree is I try to identify what these things mean. And universally on pedigrees, you're going to see circle is a female, square is a male, and a shaded square or shaded circle is somebody that has a trait, shows a trait, displays the phenotype we're looking at, displays a phenotype, right? So we're looking at a situation here in which we have a pedigree. And what I usually like to do is I draw two little lines, blanks over here, over all of these, right? Now, I want to know, when I'm looking at a pedigree, I want to know, how is this trait inherited? I see somebody is affected right here. And I'm asking myself, when I look at a pedigree, how is this trait inherited? Now, as we know, we have, there's many different ways, patterns of inheritance. Two of the main ones that I'll talk about in this video to keep it somewhat short is autosomal recessive and sex-linked recessive. Okay, so let's look at autosomal recessive first, right? We're just going to take these one at a time. If this is an autosomal recessive trait, remember, autosomal is all of your non-X and Y chromosomes, right? So this is just a general, normal, recessive trait. So could this be inherited as an autosomal recessive trait? Well, I'd like to start filling this out, right? First, I'll look at the guy that's affected. If this is a recessive trait, I'm just going to make up a letter here. We'll say A. What do you think this guy's genotype would be? And write it down in these two blanks. You can pause if you want. Hopefully you're back. You should have come up with little a. This guy's affected, right? Now let's look at these other ones. What about this guy? Is he affected? No. So let's use a big A. Do we know what the second letter is? No. This one? Unaffected. Unaffected, meaning they all are showing dominant right here. So now we look at this little a, and we say, hey, could this guy have inherited this from these parents who are unaffected? And the answer in this case is, well, it's sure, that's a possibility, right? I mean, couldn't this second letter here be little a? Couldn't he have received this little a from one parent and this little a from another parent? Sure. So if we were to say, could this be autosomal recessive? We would have to say, yes, this could be autosomal recessive. Let's look at a different one here. Let me erase my A's and my lines. Yeah. Let's look at sex-linked recessive. So for sex-linked recessive, or more specifically in this case, we'll say X-linked. X sex-linked recessive, right? In other words, the gene located on X, the X chromosome. First, the first thing I like to do is look at your males over here. Let's look at our males, because we know males all have a Y chromosome here. So I'll fill in the Ys. We know that one of their chromosomes is Y. And now for X, let's use A's again. We'll say X big A and X little a, just like that. Okay. So if there's a female that has both X little a and X little a, they would have the trait, right? We know that these females do not have the trait, right? So we know that they at least have that dominant allele, X big A, X big A. This male does not have the trait, so he has an X big A, right? This one does have the trait. She's an X little a. We know this female is not affected, so we know that she has an X big A. We know the male's not effective, so we know that this is an X big A as well. Keeping in mind that this one is the trait I'm asking about recessive. Okay, so the question is, could this person that has this disorder have received this as on an X chromosome, as an X-linked recessive trait? Well, let's look at it. We know where you got the Y from. All the Y chromosome always comes from the dad, right? But what about this other one, this X little a? Could the mother have had an X little a? Well, sure, that could have came from the mother, right? So could it be X-linked recessive? Sure. There's nothing saying that it couldn't be X-linked recessive. So let's test. Let's do one more before I end this video, so that you can kind of get the hang of this. Why don't we test autosomal dominant traits? Autosomal dominant. Autosomal dominant. Could this be an autosomal dominant trait? So let's look at autosomal dominant. Again, we're not dealing with the X and Y chromosomes anymore. We're just going to be dealing with big A and little a again. Only this time, the trait is caused by the dominant allele, right? Normal is little a. So you can think of this two different ways. First, you can look at the affected person and say, oh, they have to have a big A because they have the trait, right? Also, the normals are little a, right? And since it's recessive, all of them would have to be homozygous recessive in order to be normal, right? Anybody that doesn't have the trait. So now you ask yourself the question, could this person have received this big A for the trait from mom or dad? And the answer to that one is no, they can only give up the normal recessive trait, right? So autosomal dominant, this one could not be. This one could not be autosomal dominant. So I hope that helped you start figuring out how to work pedigrees, and I hope genetic linkage makes a little bit more sense. And I think I'm going to leave it at that for topic 5.4. Thank you for listening.